Hey everybody, we are finishing up Module 1, Lesson 3. At this time, we're going to be looking at how viruses reproduce. The main thing that we're going to be looking at is the genetic material of a virus. What is it? How do viruses infect cells? What is the general life cycle of a virus? So we're going to be looking specifically at the lytic and lysogenic cycles of viruses. We also have RNA viruses. And then what is the life cycle of COVID, SARS-CoV-2? Okay, so the genetic material of a virus is going to be DNA or RNA. With COVID, we said it's an RNA-based virus, but all organisms use DNA. And in the DNA, we have genes. So the genes inside the DNA are going to code for a single protein typically. But the sum of all the genes in an organism is called its genome. So when we look at the examples here of viruses that are DNA viruses, we have smallpox, chickenpox, and herpes. And then RNA viruses, we have some examples, HIV, influenza, and SARS-CoV-2. So viruses infect cells by using proteins on the surface of their cell, surface of the cell to get into the cell. So they'll have proteins on their surface, and then they will use that to connect with proteins on the surface of the cell. These proteins will be specific, they'll have specificity, meaning that viruses can only infect certain types of cells, which might even limit them to certain species. So in general, once the virus has landed on the surface of the target cell, it's got to get in. And then once it gets in, it is going to follow one of two pathways. So it will either follow the lytic cycle, which is where it can immediately begin using the resources of the host cell to manufacture new viruses. So it just comes in, inserts its DNA into the host. It starts making copies of that virus, of itself, basically. And then here at this phase here, maturation, you see they're assembled and then they are released. So to slow that down, the virus enters the cell here. So here's the virus. It's going to inject its genetic material into the cell. And then in the lytic cycle, it will go this way and synthesize viral components. Then those viral components are assembled into viruses. Then from there, the viruses burst out of the cell, and this often or results in the death of the host cell. The other thing it can do is integrate directly into the host genome and delay its own reproduction until a later date. We call this the lysogenic cycle. So it comes, it infects the cell, it inserts its DNA, and then that is incorporated into the host cell DNA. So this is a bacteria, and this specific type of virus here is a bacteriophage, and it's inserting its virus DNA into the host genome. From there, the bacteria replicates. So the bacteria is going to make copies of itself. And as it does that, it will also copy the viral genome that it has incorporated into itself. Then at some later time, all of these guys will flip on, I guess you could say, the viral genome. And then the cell is going to go through this process of making the viral proteins, assembling the viral components and proteins into the virus itself. And then the virus will burst out of the host cell, rupturing and killing the cell. So that is the difference between the lytic cycle versus the lysogenic cycle. These are the phases of the lytic cycle. I'm not going to read all of these to you because I don't want to bore you to tears, but you can read through them. We have the virus attaching, penetrating, putting its genetic material into the cell, and then making its own proteins and things that it needs for itself. Here in the biosynthesis phase, maturation, this is where the viruses are assembled, and then the baby viruses burst out and are released from the host cell, and this will kill the host cell. In the lysogenic cycle, again, the DNA is integrating into the host cell's DNA, but the virus will go kind of dormant or inactive. And then while this is happening, the host cell will replicate itself. Then we're going to end up with a whole bunch of host cells that have DNA in them. HIV is an example of a virus in humans that uses the lysogenic cycle. Scientists have estimated that almost 8% of the human genome is actually viral in nature, and thus evidence of how active the lysogenic cycle has been throughout our evolutionary history. So examples of RNA viruses, we have HIV, influenza, and coronavirus. The issue here is the host cells require DNA, not RNA, to start protein synthesis. So with RNA viruses, we have a couple of solutions to solve this problem. So the host cell needs DNA to transcribe into messenger RNA, and then that messenger RNA is translated into proteins. So with a virus, if it's coming in with RNA, it has to figure out either how does it get to the DNA to go from DNA to RNA, or how does it get to be RNA to be translated. Okay, so that's its issue. So the solution that is one that we see is we have HIV as a retrovirus. And before new viruses can be manufactured, the virus has to convert its RNA into DNA. So that's how it solves this problem. So HIV converts its RNA to DNA by using an enzyme called reverse transcriptase. The other solution is with influenza and the COVID virus is that they convert the viral RNA genome into messenger RNA molecule for translation. So in this scenario, they take the RNA and make it into mRNA so that it's directly translated into viral proteins. So this is what we're talking about with coronavirus. We have it attaching to these ACE2 receptors on the cell surface, and that the cell just lets it in. It's like it's a key unlocking the door, and the cell just says, "Come on in, we recognize you." When in reality, this is a thing that's been evolved by the virus to trick our cells. Once the virus gets in, it uncoats and it lets loose its viral RNA. From there, the viral RNA molecule is going to go over here to the rough endoplasmic reticulum. And it looks like they're saying, let's double check over here. All right, sorry, I lost my place. So the single-stranded RNA molecule is released into the host cell. Then for step 2, they're saying translation and replication. The RNA molecule is translated. So we're going to take the RNA and read it and see what proteins it's instructing the cell to make. So it's going to have RNA polymerase translate to convert the viral genome into mRNA, messenger RNA, and then that's going to be translated into structural proteins. So over here, we should see some structural proteins coming out of the rough endoplasmic reticulum. And then it's going to head to the Golgi apparatus for final processing. And it will be sent to the cell surface for release. So in assembly, we're going to take the structural proteins that we translated, S, M, and E. We're going to assemble the envelope and the Golgi apparatus over here. And then N proteins are used to assemble the nucleocapsid. We'll package it within a lysosome, and then the vesicle releases the virus by exocytosis from the infected cell. All right, so I'm going to let you guys answer these on your own, and that way we can get on to our next lesson.